Okay, let's jump in. We've got a fascinating set of sources today, all focused on colon cancer.
Right. A really significant health challenge worldwide.
Absolutely. And while we do have treatments, they often come with, well, some serious drawbacks.
Yeah.
Side effects, the cancer fighting back.
Resistance issues, yeah. And just getting the drugs to the tumor effectively is often tough.
Exactly. So that's our focus for this deep dive. How working at the incredibly tiny scale, the nanoscale, is offering some really innovative solutions.
We're looking at nanotechnology, and specifically nanomedicine, and how it might help us get past those limitations.
Yeah, aiming for treatments that could be, more effective, hopefully less toxic, maybe even more personalized down the line.
The sources definitely position nanomedicine as a potential game changer here. It's about manipulating materials at that near atomic level.
Creating things that interact with the body and the disease in totally new ways.
Ways that just weren't possible with older drug formulations.
So maybe let's start with the problems. What do the sources really highlight as the main issues with current colon cancer treatments?
Well, a big one is that non-specific action, especially with chemotherapy.
Right.
It's designed to kill rapidly dividing cells. That includes cancer cells, great. But it also hits other rapidly dividing cells. Hair follicles, cells lining the gut, blood cells.
Which leads directly to that systemic toxicity, right? The side effects everyone dreads.
Exactly, because you need enough drug circulating everywhere to hopefully get a high enough concentration at the tumor.
So you end up sort of poisoning healthy parts of the body too.
Which is tough on patients. It affects quality of life.
Yeah.
And the sources point out it can really impact whether people stick to their treatment plan.
Yeah, hard to keep taking something that makes you feel awful.
Definitely. And then there's the whole issue of drug resistance.
Ah, the cancer learning to fight back.
Yeah, cancer cells are incredibly adaptable. They can figure out ways to pump the drug out or change the pathways that the drug targets.
So what worked at first might just stop working.
It happens all the time. Plus, even getting the drug into the tumor can be hard. Solid tumors can be dense, poorly vascularized.
Like trying to penetrate a fortress.
It's a good analogy. So all these challenges together really underscore why we need something different.
And that's where this tiny tech comes in. Nanotechnology.
Precisely.
So working at this scale, 1 to 100 nanometers? Unbelievably small. Why is that the key? What advantages does it offer for colon cancer?
Well, there are several key things the sources focus on. First, targeted delivery.
Okay.
You can actually engineer these tiny particles, these nanocarriers, to specifically find and stick to cancer cells or the tumor area.
So like you said before, less like carpet bombing and more like a guided missile?
Exactly like that. It improves specificity dramatically, and that enables the next advantage, controlled release. Meaning you can design the nanocarrier so it only releases its drug payload when it gets to the tumor environment.
How does it know? What triggers it?
It can be triggered by conditions unique to the tumor. Like, tumors are often slightly more acidic than healthy tissue, or they might overproduce certain enzymes. The nanoparticle can be designed to react to that specific pH or those enzymes.
A tiny smart bomb, basically, waiting for the signal.
That's the idea. Very clever.
And there's this other concept mentioned, the EPR effect, enhanced permeability and retention.
Yeah.
How does that fit?
Right, EPR is really important. Tumors tend to grow fast and build messy, leaky blood vessels. Nanoparticles are small enough to slip through those leaks into the tumor tissue, but then, and this is key, they get trapped.
Trapped? Why?
Because tumors usually have poor lymphatic drainage. Stuff gets in easily through the leaky vessels, but it doesn't get cleared out efficiently like it would in healthy tissue.
Wow. Okay, so the tumor's own flaws, the leaky vessels, the bad plumbing, actually help the nanoparticles build up right there.
It sounds strange, but yes, it's a property nanomedicine exploits. Healthy tissues don't trap the nanoparticles nearly as much.
Which directly helps reduce that systemic toxicity we talked about.
Huge potential there. You get more drug at the tumor, less circulating elsewhere, causing side effects.
Makes sense. And the sources also mentioned multifunctionality, doing more than one thing.
Yes, that's another exciting aspect. These nanoparticles can often be engineered to carry multiple drugs at once.
Or even combine treatment with diagnosis.
Exactly. That's theranostics. Imagine delivering a drug and an imaging agent in the same particle.
So you could potentially see where the drug is going, maybe even monitor the response in real time.
That's the goal, a level of feedback and precision we just don't have with most current treatments.
Okay, so the why is clear targeting, controlled release, EPR, less toxicity, multifunctionality. Let's talk about the how. What kinds of nanoparticles are being used? The sources mentioned quite a few.
Yeah, think of them as different kinds of delivery vehicles. One common type is liposomes.
Tiny fat bubbles, basically, like cell membranes.
Pretty much. Made of lipids, very biocompatible. They can carry different drug types, water-loving, fat-loving, good for sustained release.
Okay.
The sources mention liposomal 5-fluorosil as an example being looked at for colon cancer to keep drug levels steady longer.
What else?
Polymeric nanoparticles are big too. Made from plastics, usually biodegradable ones that break down safely.
Versatile.
Very. You can tweak their size, how fast they break down, and crucially, you can stick targeting molecules onto their surface.
Putting that address label on them.
Exactly. Then you have dendrimers. These are really interesting, highly branched, sort of tree-like structures.
Tree-like?
Yeah, lots of branches mean lots of places on the surface to attach things, multiple drugs, maybe genetic material like Suriname to, say, shut down resistance genes.
So they can carry mixed cargo?
Yeah.
Any metals involved?
Yep, metallic nanoparticles, gold, iron oxide are common examples. They can carry drugs, but also have their own therapeutic uses. Like generating heat when you shine light on them, that's photothermal therapy. And they're great for imaging too. Theranostics again.
Got it. And silica?
Mesoporous silica nanoparticles. Imagine tiny sponges with lots of ordered microscopic holes.
Okay.
That structure gives them a huge surface area to load up lots of drug, and you can control the release from those pores quite well.
Like a very precise sponge.
Yeah.
And the last one mentioned was?
Layered double hydroxides, or LDHs. These are inorganic layered materials.
What's special about them for the colon?
Their pH sensitivity could be really useful, especially for oral delivery. The pH changes a lot as things move through the gut, and LDHs can be designed to release drugs in response to those specific changes. They can also carry drugs and things like DNA or RNA between their layers.
That is quite a range of options.
Yeah.
So with all these carriers, how are they actually directed? You mentioned active targeting.
Right. So beyond just relying on that passive EPR effect, active targeting means putting specific keys on the nanoparticle surface.
Keys that fit locks predominantly found on the cancer cells.
Exactly. These locks could be receptors or proteins that are much more abundant on colon cancer cells or maybe on the tumor's blood vessels than on normal cells.
Examples?
Things like folic acid receptors, certain proteins like EpCAM, specific sugar-binding molecules called lectins, or peptides that stick to tumor blood vessels. It gives an extra layer of specificity.
Okay, and then the triggering those stimuli-responsive systems.
Yeah.
Making the smart bomb go off.
Yeah, that's about responding to the local environment. We mentioned pH releasing the drug in the slightly acidic tumor area.
Right.
Also, enzyme-responsive systems, specific enzymes that the tumor turns out, can break down part of the nanoparticle and release the drug.
So the cancer's own activity triggers its destruction.
That's the elegance of it. Other triggers being explored are things like temperature changes, maybe induced from outside, or differences in the chemical environment, the redox state, inside tumors versus normal tissue.
It all aims for release only at the target site.
Minimizing collateral damage.
And this precision seems ideal for combination therapy, right? Hitting the cancer with multiple things at once.
Absolutely. Packaging different agents in one nanoparticle ensures they arrive at the cancer cell together in the right proportions.
For a synergistic effect. A 1-2 punch, or even more.
Precisely. You could combine, say, a standard chemo drug with something that boosts the immune system against the cancer. That's chemo plus immunotherapy.
Okay.
Or chemo plus Suriname to block drug resistance mechanisms. Or even chemo combined with those light-activated therapies, using the nanoparticle to help generate heat or reactive oxygen to kill the cells.
A coordinated attack. It sounds incredibly powerful on paper.
Mm-hmm.
Where are we in reality? Is any of this actually being used in patients yet?
Well, the sources show a lot of strong preclinical data. Lab studies, animal models. They're showing increased effectiveness against tumors and definitely reduced toxicity compared to the free drugs.
Promising signs. But human trials.
Some are moving forward. It's not widespread standard care for colon cancer yet. These specific nanomedicines are still largely in development phases.
But the concepts are being tested.
Yes. Formulations like nanoparticle albumin-bound paclitaxel nab-paclitaxel, originally for other cancers, are being looked at. Liposomal irinotecan is another one that's progressed into clinical trials for advanced colorectal cancer. So it's happening, but it's a long, complex road from the lab bench to the clinic.
If this continues, though, what's the big picture hope? The future vision?
The sources paint a picture of truly personalized medicine. tailoring treatments not just to colon cancer, but to the specific molecular details of your tumor using these targeted nanocarriers.
And using their diagnostics for real-time adjustments.
Potentially, yes, monitoring response, tweaking doses, and ultimately the dream is less invasive, more tolerable therapies, maybe even outpatient treatments that significantly improve quality of life.
It sounds fantastic, but it's never that simple, is it? What are the major roadblocks still ahead? The sources must-mention challenges.
Oh, absolutely. They're realistic. A big one is just the regulatory pathway. How do agencies like the FDA evaluate these complex new nanomedicines? The frameworks are still evolving.
They don't fit the old mold.
Not easily. Then there's manufacturing.
Right.
Making these intricate nanoparticles consistently, reliably, and affordably at large scale is a huge technical and economic challenge.
And safety. Injecting novel materials. What about the long term?
Crucial question. We need more long-term data on biocompatibility and potential accumulation or toxicity over years, not just weeks or months.
Right.
And finally, cancer itself is variable. Tumors differ between patients, even within the same patient.
So a targeted approach might work great for one person, but less well for another.
That heterogeneity is a real challenge. How well the targets are expressed, the tumor microenvironment, it can all affect how well these nanodrugs work, making consistent success across all patients difficult.
Okay, so pulling this all together, the main takeaway from this deep dive.
I'd say it's that nanomedicine represents a fundamental shift. It offers tools to potentially make colon cancer treatment much more precise, more effective, and critically less toxic than what we often rely on now.
Holding out hope for really significant improvements in how patients fare, their outcomes, their quality of life.
Definitely. But as the sources make clear, there's this gap between the incredible science happening in the lab and getting these treatments approved, affordable, and working reliably for everyone. Bridging that gap is the next major hurdle.
Which really leaves us with something to think about, doesn't it? Considering this amazing sophistication at the nanoscale, what is that gap between the cutting-edge innovation and the real-world hurdles of cost, regulation, long-term safety, patient variability? Tell us about the path forward for cancer care overall.